Hey guys, welcome back to my channel One Stop Biology. Basically yesterday we studied the first chapter of NCERT Class 11th and today we are going to start the second chapter, that is biological classification. Okay, so before we start the chapter, I hope you would have gone through the chapter 1 video. If not, please go and watch it once. Also, if you want to listen to the same video in Hindi, you can go and check the channel that I've created, wherein I'll be putting all the videos in Hindi as well, the link of which is in description of this video. So let's start with the chapter, which is biological classification, and this is chapter 2 of NCERT Class 11th. Okay, I also hope you are accessing the notes provided by me, which are basically detailed notes on my website onestopbiology.com. Okay, so you can access all the notes provided by me. So what is biological classification? As we also studied in the previous chapter, that in order to keep it simple, in order to make a hierarchy of all the organisms, you classify them biologically and you keep them in separate groups. So for the very first time, Carolus Linnaeus gave two kingdom classification, wherein he divided all the living organisms into two, basically one being kingdom Plantae and the other being kingdom Animalia. So all the plants, as in all the organisms which are grouped as plants were kept in Plantae, and all the organisms which are considered to be an animal were kept in kingdom Animalia. However, Carolus Linnaeus doesn't distinguish between what? So he doesn't distinguish between eukaryotes and prokaryotes. He doesn't distinguish between unicellular and multicellular organisms and photosynthetic and non-photosynthetic organisms. Okay, so he just gave a very simple classification, which was two kingdom classification. One was plant and the other was animal. The very important classification given later on in the year 1969 was by R.H. Whittaker. So R.H. Whittaker, what he did was he proposed a 5 kingdom classification. Remember that he proposed a 5 kingdom classification, and what he did basically was he divided these organisms into five different groups, starting with the first being Monera, then Protista, fungi, Plantae and Animalia. So these two were the same as given by Carolus Linnaeus, and he added 3 more: Monera, Protista and fungi. So what are the criteria for this classification? The criteria of this classification were cell structure, thallus organization, we'll study about what thallus is, mode of nutrition of these species, their reproduction cycle, and the phylogenetic relationship of them. Okay, so these are various criteria for classification. Now, what exactly are the characteristics in details? We'll see that as well. So there are certain detailed characteristics. Okay, so let's see this chart given in the NCERT chapter. So what does it say? So it says a lot about different kinds of structure or organization of these organisms, which are divided into 5 groups. What are these characters? These characters are cell type. Okay, so either they can be unicellular or multicellular, so it can be prokaryotic or eukaryotic. So as you can see, only Monera, only the kingdom Monera is prokaryotic. Rest all the other kingdoms are eukaryotic. So all the organisms of the later 4 kingdoms are eukaryotic. Then we come to cell wall. So on the basis of cell wall, again It is present in some and absent in some. If you see here, Monera is non-cellular, so they have a cell wall of polysaccharide and amino acid. But it's not exactly how we study the cell wall, it's non-cellular. Now in protista again it is present in some. In fungi it is present but it is without cellulose. Plants are considered to have a true cell wall which is made of cellulose, and in animals we basically lack cell wall. Then comes nuclear membrane. Again, nuclear membrane is absent in Monera but present in all the other 4 kingdoms. Then it comes to body organization. So whether your body organization is unicellular or multicellular. So monerans and protistans are cellular, while fungi are multicellular but loose tissue organization, and plants and animals have basically highly developed body organization, which is tissue and organ or organ system as well in case of animals. Okay, now what is the mode of nutrition for them? So whether they can prepare their own food and they are autotrophic, or they are dependent on someone else, which is heterotrophic. So monerans basically are autotrophic as well as heterotrophic. So they can prepare their own food. They can basically they involve the process of photosynthesis, and in some case they can depend on another organism. They can be a saprophyte or parasite. In case of protistans, it is again autotrophic and heterotrophic both. In case of fungi, they cannot produce their own food. They are only and only heterotrophic. In case of plants, it's the exact opposite. They can prepare their own food. So all the plants that exists on earth can prepare their own food by the process of photosynthesis. And then animals, again the exact opposite of plants. So animals are solely dependent on other living organisms, be it plants or animals, for their nutrition, and hence they are heterotrophic. So these are the 5 kingdoms and basic characters that the organisms of these kingdoms own. Okay, so we will then, now what we'll do is we'll start with the first kingdom, which is kingdom Monera. Now what exactly this kingdom is? So all the bacterias, remember that all the bacterias are sole member of these kingdom, and they are the most abundant microorganisms. Please remember that bacteria are the sole member and they are most abundant in nature. So what happens with them is they basically can survive in or they live in extreme habitats. Like they can survive in hot springs, they can survive in deserts, snows, deep oceans, and in other organisms as parasites. So we have a lot of bacteria as parasites in humans as well. Now how we group it? So basis their shape, we basically group them into 4 different categories, and these are the 4 shapes. Okay, so as you can see, there are certain demarcations in this shape. You can I think you can actually tell me what are the shapes. So the first one, as you can see, is nothing but a spherical shape, right? So this is the spherical shape. Then so this is basically the first that you can see, coccus bacteria. Okay, then the second one is nothing but rod shaped. These are bacilli. Okay, these are rod shaped and they bear spore as well. Okay, so these are rod shaped bacillus. Third one are basically spiral in shape, as if you can see here, these are spiral in shape. So these are spirilla, and they also have flagellum. Okay, so these are spiral in shape. So these are basically spirillum. And the last one are somewhat how we put a comma. If you remember, we put a comma like this, right? So these are comma shaped and they are known as Vibrio. So these are comma-shaped Vibrium. So on the basis of shape, you can group bacteria into 4 categories. So the first one being spherical is coccus or cocci in case of plural. So the first one being spherical coccus, second one being rod-shaped bacillus, third one being comma-shaped Vibrium, and fourth one being spiral spirulum. Okay, these are some characteristic features of these two bacteria, wherein one owns flagellum and the other one owns a spore. Okay, so what we are going to do after this is we are going to study some more forms of bacteria in detail. So we'll start off with the first one. Okay, so we are going to start with Archaebacteria. Okay, so basically Archaebacteria are nothing but there's one characteristic feature of these bacteria is that their cell wall is made of muric acid. Okay, remember this feature, it is important from exam point of view. So in case of Archaebacteria, the cell wall is made of muric acid. You can see it's mentioned here. Okay, so these basically live in some of the most harsh habitats such as extreme salt areas. If they live in salt areas, they are known as halophiles. In case of hot springs, they are known as thermoacidophiles. And in case of marshy areas, they are known as methanogens. Okay, so how they are different? As I said, they are different from other bacteria in having a different cell wall structure which is made of muric acid, and it is basically responsible for their survival in extreme conditions. Okay, so we'll see some features of these different types of bacteria as in different type of Archaebacteria. So basically, what are methanogens? Methanogens are present in the gut of several animals such as cows and buffaloes. And what they're responsible for? They're responsible for production of methane, biogas from dung of these animals. Okay, so methanogens, what they do is they produce methane in certain animals, and you basically get methane from their dung. Okay, now there are some basically they are the most primitive prokaryotes. They are the most primitive prokaryotes, and absence of peptidoglycan in cell wall. So again, this is one important property, you should remember it, that there is absence of peptidoglycan in cell wall. Okay, the third point is they have monolayer of branched chain lipid in cell membrane. So we are basically considering various different characteristics of Archaebacteria. So there are different kinds of Archaebacteria, how they are different and why they survive in very harsh or most harsh habitats. It's because of cell wall is made of muric acid, which basically protects them. Then the second feature is that they are the most primitive ones, and the absence of peptidoglycan in the cell wall. Okay, now what is the next point? So next point is they have a monolayer of branched chain lipids in cell membrane. Okay, so they have monolayer of branched chain lipids. What is the next? They are obligate anaerobes or they are facultative anaerobes. Okay, so remember these 2 points as well. So they are either obligate anaerobes or they are facultative anaerobes. Okay, the last point being since they are so primitive, they are also known as living fossils. They are also known as living fossils. Okay, so here we are basically ending the Archaebacteria thing, and we will be starting with the second kind of bacteria, which is eubacteria, and which is also known as true bacteria. Okay, so just let me clear my screen. Yes. So what are eubacteria or true bacteria, and why they are considered as true bacteria? Because they have a rigid cell wall. And if they are motile, they have a flagellum as well. Okay, so they have different kinds of bacteria. One being cyanobacteria, which is also known as blue-green algae, and they have chlorophyll A, which is similar to green plants, and they are photosynthetic autotrophs, so they can prepare their own food. Okay, now what is the characteristic features of eubacteria? They are unicellular, they are colonial or filamentous, freshwater and marine both. They can be found as terrestrial algae. They are generally surrounded by gelatinous sheath. Okay, so please remember all these points. Okay, now the next point is they form blooms in polluted water bodies, and they can fix atmospheric nitrogen in specialized cells, which are known as heterocysts. So what basically they are trying to say is they basically form blooms and in polluted water bodies. So you will see in very old lakes where the water is being there for a longer time, you will find a lot of blooms of these bacteria, and what they do is they can fix atmospheric nitrogen in specialized cells, which are known as heterocysts. And here you will find an example of eubacteria, which is nostoc. Okay, so this is nothing but a diagram of nostoc, which is a filamentous blue-green algae. Okay, so here the structure that you see is nothing but heterocyst. Okay, and they have a mucilaginous sheath above the structure. Okay, and this heterocyst is responsible for fixing atmospheric nitrogen basically. Okay, then comes chemosynthetic autotrophic bacteria. So chemosynthetic autotrophic bacteria basically oxidize various inorganic substances such as nitrates, nitrites, ammonia, and they release these energies in the form of ATP production. Okay, and that is why they play a great role in recycling nutrients. So chemosynthetic autotrophic bacteria oxidize various organic substances and produce ATP, produce energy. They release energy, and that is why they play a great role in recycling nutrients like nitrogen, phosphorus, iron, and sulfur. Okay, this is something about chemoautotrophic bacteria. Then we'll go to heterotrophic bacteria. Now what are these bacteria basically? So heterotrophic bacteria are the most abundant one in nature, and they are very important. Always remember, they are very important decomposers. Okay, so they basically help in making curd from milk. Okay, they help in production of antibiotics. They fix nitrogen in legume roots. Some are found as pathogens as well in human beings. So all the bacteria that are found in humans as pathogens are nothing but heterotrophic bacteria. And the diseases that you hear about, like cholera, typhoid, tetanus, right? So these diseases are caused by these heterotrophic bacteria. Okay, so basically how does these bacteria reproduce? They basically reproduce by fission, and they produce spores under unfavorable conditions. So if the condition is not favorable, basically if the condition is not favorable for a bacteria to reproduce, what they do is they form spores. And once they get a favorable condition, they start reproduction by fission. Okay, and in some cases they reproduce through sexual reproduction as well, which is again what they do is they basically transfer DNA from one bacterium to another. Now here you can see an example of nothing but a dividing bacterium. Okay, so this is a dividing bacterium. And what are the basic parts of the bacteria? One is the cell wall. So the outer covering is the cell wall. Inside it is the cell membrane. Yes, you got it correct. And inside that we have the DNA, which they transfer from one bacteria to another. So how they reproduce is basically so one bacteria divides through fission, and what they do is they divide they transfer the nuclear material, which is DNA as well, from one part to the other part and becomes two bacteria. Okay, so this is the reproduction of bacteria. In this part of the second chapter, basically we have covered what all things. We have covered the kind of classification, which is 2 and 5 kingdom classification. Then what we covered, we started covering the first kingdom given in 5 kingdom classification, which is Monera. And Monera is nothing but all kinds of bacteria, and we started off with the bacteria. Now in again bacterias, we saw what kind of bacteria are there depending upon the shape, their shape, and then we charted out some other forms of bacteria. So we studied Archaebacteria, we studied Eubacteria. Okay, and we then divided these groups as well into different other forms. Okay, there's just one more thing left, which is Mycoplasma. Okay, so what is Mycoplasma? Basically, Mycoplasma is nothing but organisms that completely lack the cell wall. Remember, so they completely lack the cell wall, and they are the smallest living cell known, and they can survive without oxygen. Okay, so these are organisms that completely lack a cell wall. They completely lack a cell wall. They are the smallest known, and they can survive without oxygen. And they again are pathogenic in animals and plants. A lot of Mycoplasma are pathogenic in animals and plants. So with this, we come to an end of the first kingdom of 5 kingdom classification given by Whittaker, which is kingdom Monera. Do let me know if you have any doubts after going through the video or after going through my notes given on my website One Stop Biology. You can ask me questions in comments here, or you can even post your doubts on the WhatsApp number given in the description box of this video. Do let me know if you have any kind of suggestions for how I should improve, or how I should make you understand more and more with every coming videos. I hope you like my video, and if you do, please share it with your friends. Tell them that it's completely free, and I am going to cover the entire NCERT syllabus before your boards and NEET exam. There's nothing to worry, and thank you for watching this video. Please do subscribe to this channel in order to get recent or daily updates. Thank you so much. Bye bye.